guys, welcome back. Welcome back to another video from Learning with Isha. What are we discussing today? Biomolecules, okay? What are biomolecules? Well, biomolecules are all carbon-based compounds. They are all carbon-based compounds found in a living tissue. Okay, but for that we need to understand what a living tissue is made up of. Well, any tissue, any cell that eventually forms an organ system that makes an organism is made up of, on the basic level, elements and compounds. Okay, elements and compounds. So what are elements and compounds? Well, elements are those substances that cannot be broken down into smaller units. Like if I have a calcium ion, I would not be able to break it down any further, right? But now if I make a compound, calcium chloride, right? I can break this down into calcium ions and chlorine ions, right? So this is a compound, this is an element, they come together, form other compounds, and then what is made eventually an organism. So the study of biomolecules is actually the molecular level study of what is happening inside the organism, right? We study about proteins, we study about enzymes, we study about the various processes that happen in the human body. But what are the molecules that are causing the process, right? If hemoglobin, right, it carries oxygen. I need to understand hemoglobin is a protein, right? But to understand that, I need to know what is a protein. So in this chapter, it is the story of that, and today is the introduction of this chapter. So now we begin this chapter with the very basic question that how do I know what are the kind of elements and compounds found in the human system or in any living system for that matter. Let's say a bacterium, a fungi, a plant, they're all made up of biomolecules, carbon-based compounds, right? So various experiments, various analysis were done to understand that. So first what scientists thought was, okay fine, let me compare living tissues. Let me compare living tissues to the elements found in the Earth's crust, right? To the elements in Earth's crust. Okay, so what is the Earth's crust made up of? It's made up of many elements, see guys. It has carbon, hydrogen, oxygen. But you'll be very surprised to know that the same elements that were found in the Earth's crust were also detected in the human body. Isn't it very striking? Yes. What is furthermore striking is that there were only two elements whose quantity was more in the human body than found in the Earth's crust, like percent weight wise. Okay? The second analysis was to analyze organic compounds in any living system. So how do I do that, right? Okay, let me give you an example. Let's say we have a leaf, okay? I take a small portion of the leaf and I try to analyze what are the biomolecules that are present in the small portion of the leaf. Okay, let me take a small piece. Now what I will do is, I'm going to take the small piece and grind it, okay? Grind it using a mortar and pestle. Okay, what is a mortar and pestle? जब चाय बनती है तो मम्मी कूटती है ना अदरक को, किस चीज से कूटती है? Using a mortar and pestle. Okay, that is a mortar and pestle. Now अगर मुझे किसी चीज को कूटना है तो मुझे क्या चाहिए? कोई solvent भी चाहिए, right? So that solvent is it's a compound known as trichloroacetic acid, right? What is acetic acid, guys? CHHHCOOH, right? Now what I'm going to do is substitute these three hydrogens with chlorine. What do I get? Trichloroacetic acid, right? So now I'm going to grind this, and what I'm going to get out of it is a slurry. I get a slurry. So what is a slurry? See guys, a slurry is a thick paste, okay? When I grind a tissue, that could be, let's say, a leaf in this case, a piece of liver, a piece of meat, right? Whatever I'm using for my study, I'm going to grind it and I will get a thick paste out of it, fine? Now, what I'm going to do in the next step is going to strain it using cotton cloth, okay? I'm going to take a cotton cloth and strain it out. Like, घर में पनीर बनती है तो क्या होता है? दूध को फाड़ के उसको पूरे क्लॉथ से छान देते हैं. Exactly यही इस पेस्ट को हमें छानना है. We have to just filter it out, right? So जब हम चाय छानते हैं, for example, तो चाय छन्नी में क्या होता है? एक लिक्विड आ जाता है, the tea comes out, and what I have left is the tea leaves, right? Or whatever I have put in the tea. Okay? So now similarly, I will get two portions. First will be called the filtrate. Second would be called the retentate. Okay. See, what does retention mean? Retention means to hold back, right? So the cloth is going to hold back a portion which will be known as the retentate. And the portion that will get dissolved with the trichloroacetic acid and come down in my collection bowl will be the filtrate, right? This is my collection bowl. This is my cotton cloth. I have put the liquid. What stays back over here is what my retentate. What comes down with the trichloroacetic acid is the filtrate. So what will naturally come down with the trichloroacetic acid has to be soluble in it, right? If you make tea and you add sugar to it, right? So the sugar is soluble in my tea. That is why it will come down in my cup. But the tea leaves are not soluble in the tea, so they are going to stay put, right? So now let's see the differences between filtrate and retentate and how it is going to help me. Because my main aim was not to create this filtrate and retentate, right? My main aim was to analyze organic compounds. So the filtrate, since it is dissolving in the trichloroacetic acid, it's going to be called the acid soluble fraction. Okay, it will be called the acid soluble fraction, and this will be called the acid insoluble fraction, right? It is not soluble in the acid, it is staying back. Acid insoluble fraction. Naturally, the sugar will be smaller in size than the tea leaves, right? So this one is going to be, these molecules that come down will be of a smaller size, right? 18 to 800 Daltons. And the retentate molecules are greater than 10,000 Daltons in size. Okay? So these small molecules are called micromolecules. They are called micromolecules. And these larger molecules are called macromolecules. Okay? Micro means what? Micro means small. Okay? Macro means big. Okay? So small molecules will be called micromolecules. The larger ones will be called macromolecules. So what kind of molecules stay back? Right? They are normally proteins, nucleic acids, lipids, and polysaccharides. So they are basically only of these four types. You will be surprised to know that the size of lipids is not as much. But why is it staying back then? Because normally lipids form what? The cell membrane. So when we are grinding our living tissue, the lipid will stay back, right? So it will not come down. Now talking about macromolecules, they represent the cytoplasmic content of the cell, right? Like what? They represent the water content of the cell, or let's say they represent the small amino acids, right? Or any other small compounds that might be formed in the cell. So they are micromolecules, they are macromolecules. So this analysis told me about organic compounds. Now scientists did another experiment to find out about what inorganic compounds were present in the cell. What inorganic compounds were present. Okay, fine. But now this one, an experiment done for the inorganic compounds, was a rather destructive experiment. Why is it destructive? Well, we'll see. In the first step, what we did, again we're going to take the living tissue that we want to analyze. So I'm taking my living tissue. Okay, what is this? My leaf, liver, meat, whatever it is. Right? Now, what I'm going to do is, the living tissue has some moisture content in it, right? So, I don't want to know what weight is contributed by water. I want to know what weight is contributed by the elements that cannot be broken down. So, a dehydration step is done over here, okay? So, we allow the water to be removed. So the weight of the living tissue with the water present is called the wet weight, and the weight of the living tissue minus the water is called the dry weight. Okay, so the dry weight will be less because the weight of water will get deducted now, right? So what I get is the dry weight now. In the next step, what I'm going to do, I'm going to burn this. Okay, so in the presence of oxygen, I'm going to burn this organic compound. Okay, and what I'm going to get is ash. Whenever you burn wood or coal, what is left behind is ash. Okay, that whitish powder that we see, that is ash. Okay, ash represents what? The inorganic compounds present. It represents the inorganic compounds in cell. Okay, so now this ash will tell me, an analysis of this will tell me about the elements present in the cell. Fine? Okay, now an example of this reaction, right? I'll give you a very good example. Let's say glucose is a molecule. Okay, C6H12O6 is what? Glucose. I'm burning it, so I'm adding oxygen. Okay, what will I get? Carbon dioxide and water. If you must be noting this equation, it must ring a bell in your mind. This equation is not balanced. We need to balance it, okay? It's always important to balance the equations. So now it's a balanced equation. So this is called the oxidation of glucose. So now what is going to happen? All the gaseous, see guys, we're doing an elemental analysis, okay? The significance of the burning step is that these gases will get evolved. So see, what are we practically left with and what we have excluded? We have done away with the water. We have done away with gases. Now we are left with elements, right? So this is the aim of our analysis, okay? So this is how people did experiments to find out what biomolecules were found in the living system. So the analysis was done on two levels, on an elemental level, on a compound level. An elemental level gives me, let's say I have sulfur, phosphorus, what are the things in my cell. A compound analysis is going to tell me what compounds are present in the cell. Now that sulfur is going to go, and where is it going to get absorbed in the cell? Let's say there's an amino acid, cysteine and methionine. These are amino acids, they make up proteins, right? The sulfur will go and get incorporated in these amino acids. So our attempt was to go backwards, right? We know the amino acids. Now we're trying to find out what are the elements making them up, okay? So that's all for today's video. Let's meet in our next video where I'm going to start talking about amino acids. Thank you for today.